It is. Yeah. I really did screw up. Why didn't I? Should have said, yeah, we have to be on Monday. So I should have said, isn't it? Yeah. Next, if I ever organise another one, I'll remember that. Okay, everybody. So we're going to continue with the sort of statistics theme and time series, but more of a sort of statistical modelling element. I think that's correct, isn't it, Leo? So Leo is another person I've known for a long time. I think we met in Izmir, Copenhagen, back when actually there was this horrible debate about enterotypes. Did they exist or not? And yeah, poor Leo was caught in the middle of that in his session. And you were also at Jerome Rees's lab, wasn't it, for a little bit as a postdoc, I think? Yeah, and now you're at the University of Turku running an excellent group looking at statistical modelling of microbiomes. So thank you, Leo.
Okay, thank you very much, Chris, for this kind introduction. And hello, everybody from my side. It has been a long week, but nice to see that you still have a lot of energy to follow all these interesting talks. It has been a really great program. I don't know if it's statistical, mathematical, or what. I'm a computational scientist, and we tend to blend different approaches to understand microbiome variation, and mostly in human populations, but sometimes also in synthetic communities and animal models. And I was planning to discuss and give some examples about our relatively recent work on microbiome time series analysis, especially. So I will start with some background on prospective and longitudinal population cohort studies. This is an important aspect of our work and motivation also for this analysis of the dynamics. Although often the population cohort studies currently don't really have a very rich time series, but they are still important for understanding the dynamics in other ways. Then in the second part of the talk, I will talk about the role of ecological memory in interaction models of microbial communities. I had a shorter talk about this topic in ISMA this year, and then in the end, if we have time, I will briefly touch upon some tool development that we are doing in this area for R and Julia. So the population cohort study that I mentioned is a Finnish population cohort study called FINRISK. It is a prospective population cohort study. The samples are from 2002. National Institute for Health and Welfare in Finland collected fecal samples from more than 7,000 individuals 20 years ago, and they were stored in a freezer for a long time. And then some years ago, we decided that it's time to sequence these samples. This was done in collaboration with Rob Knight, who had kindly provided the sequencing for us for the Finnish samples. So this is a representative population cohort study in the sense that these individuals have been randomly selected from Finland by kind of random population sampling, and then 10,000 individuals were invited to join, and there was quite good participation rate. They are from different areas of Finland, so we can also study some of the spatial variations along the geography in Finland. Then these are mainly middle-aged adults. Average age is roughly 50 years, and then they have the typical BMI distribution that we have in the population. We have done shallow shotgun metagenomics for this, about 1 million reads per sample. That is not really good for functional profiling, but it is sufficient for taxonomic profiling of these communities. And in Finland, we have the particular thing that is common to other Nordic countries as well. We have very comprehensive population registers, so we can follow up what happens for these individuals over time in terms of different incident diseases, deaths and other things. We also have a good demographic characterization for these individuals. We know the income levels and prescribed and purchased medications and so forth. And now, after 15 to 20 years after sample collection, about 10% of this population has died. And one of the first things we wanted to look at was whether gut microbiome at a certain point of time will predict mortality risk. And here is the PKA plot or MDS ordination for this data. So the 7,000 Finnish individuals are here on this map, and the proximity of the individuals on the map shows the similarity of the microbial community composition. I'm sure you know how to read these MDS plots. And now one thing we can see in the Finnish population distribution is that there is kind of two clouds. This is individuals with high prevotella, the ones highlighted by green. I'm showing the dominant genus by color here. And then we have a Bacteroides by red, Bacteroides dominated individual, and there is a gradient towards other taxa. And one thing that is visible here is the group of black points in the middle. That is something you see better in third BC axis. And those individuals are dominated by members of the Enterobacteriaceae family, and they also tend to die more often in the next 15 to 20 years. This is the cumulative death rates, and we can calculate the Cox proportional hazard model to just quantify the association with the third BCA axis and mortality rates. So this was one of the first things we were looking at. We also checked this from complementary angles. We were identifying co-abundance networks of the microbiome with some compositionality aware methods, and we recovered some tight clusters of microbial taxa that tend to co-occur, and we essentially recovered the same pattern here that is associated with mortality. So those individuals who have high levels of this bacteria tend to die more often. And then this association is also robust in Eastern and Western Finnish subpopulation, which is good news because in Finland we have genetical differences between East and West, and also cultural differences in terms of diet and other things. So these populations are different, and we see quite robustly this signal in these two different populations. This doesn't happen for all endpoints. For instance, in the context of liver disease, we have seen that the predictions of microbial community versus incident liver disease do not generalize that well between regions. Every region seems to have a little bit different sets of taxa that are predicting the disease. So it may depend on the endpoint. We were also looking at specific causes of death, and then it is maybe not that surprising that gastrointestinal causes of death are well predicted by gut microbiome, but then there are also other causes of death like respiratory diseases, cancers that also have significant predictive power from the microbiome. So there are different endpoints. We started with mortality risk, but we can now look at many other incident diseases in the population, and we are doing this with collaboration with others. We have analyzed liver disease and type 2 diabetes so far, and now we just launched a dream challenge on predicting heart failure based on gut microbiome. We have, based on some initial checks we did, we know there is predictive signal in the data, but this is now a competition where anybody can basically register to try if they have a prediction algorithm that can predict heart failure 15 to 20 years after the sample collection. So if you're interested, you can go to this address and register for this. We already have, I think, 70 registrants. So that's about FINRISK, but why I was talking about this population cohort study that is mostly cross-sectional. We only have one time point from 2002 in FINRISK currently. We are planning to maybe have a new sample collection next year. This would allow us to analyze the stability of the microbiota in 20-year time span, but this is ongoing work. But in general, we would like to know and understand how individuals are moving along this population landscape. So whether some of these states, let's say high prevotella, low prevotella, or some of these other states, if they are resilient and how resilient they are, how often do they switch between each other, and what's the relation between individuals versus the population level variation. So for sure there will be a lot of individual dynamics going on there, but there are also features that are probably shared by many individuals in the population, and that's why we see this population level structure also in our cross-sectional data. But one problem with these population studies is that we typically have very limited time series. We only have a few time points per individual, and typically not that many individuals necessarily. So we have much larger background collection of maybe thousands of individuals, and then we can couple this with more limited time series from a smaller number of individuals. When we combine and aggregate these two things, we can start to characterize the landscape of population dynamics. which would not be accessible otherwise. So some time ago, we were doing this for gut microbiota from the angle that we wanted to decompose the broader ecosystem into more limited sub-ecosystems. And we were showing that the gut microbiota seems to have relatively independent sub-ecosystems that show B-stable variation and do not correlate very strongly. This was a study we published in Nature Communications some years ago. We also noticed that between these B-stable taxa that we have there are notable differences in their stability properties. So Prevotella is very stable in terms of its high and low abundance states, and it very seldom shifts between those states. Then there are some other groups like Dialister that also have a tendency to have resilient states, but they switch more often. So there are differences in different parts of the ecosystem in terms of these resilience and stability properties. And we can see that already by univariate modeling approach, we can capture some of these interesting dynamics in the sub-communities. So we do not necessarily have to start with multivariate models for these community dynamics. We can start by univariate analysis. So one of these ways to measure resilience in univariate system is something we published recently in collaboration with Martin Scheffer, which looks at the drift and diffusion landscape of univariate dynamical systems. So if you assume that you have a good time series available and you can observe the fluctuations of the system along its potential landscape, then by measuring these fluctuations, we can estimate the exit times from the given stable state. And one aspect of this study is that depending on where you are in the potential landscape, the expected exit time is different. So if you are really in the middle of the potential well, then the exit times is expected to be longer than if you are closer to the tipping point. But we can integrate across these different areas on the potential valley and get a robust estimate of the exit time from this state. And this provides a new measure of ecological resilience. And as part of this, we are using non-parametric estimates of drift and diffusion. So we can estimate the drift of the system at all the different states of the x-axis. Here it could refer, for instance, to the abundance of the microbe. So with low abundance states, the abundance tends to get higher, so the drift is positive. When you get on the other side of this potential well, then the drift turns to be negative. So that's kind of what we expect to have in this kind of system that is having a stable state here. On the other hand, then when we are closer to the tipping point, then the drift goes in ways that to the left from the tipping point, you tend to return to the left, to the right of the tipping point, you tend to return to the right, and so forth. So this is a non-parametric estimate of the drift and diffusion that we were using in this paper, but it depends on the availability of these long and dense time series, which are not often available in the context of gut microbiota population studies. So we have been interested lately in the question on how to combine and aggregate information across many short time series. So instead of one single continuous time series, we could have a set of short time series. In our case, we have, let's say, 77 individuals with 2 to 5 time points each. But if we expect that all these individuals have some universal features of their dynamics of the gut microbiota, then it would make sense to look at these collectively and ask what is the shared information in all these. So we assume that these all come and reflect the same dynamical process in the gut microbiota, and then we have been developing ways to aggregate the information and characterize the potential landscape. So Chandra Ross, who also had a poster here and has been around here, maybe some of you saw his poster, has been working on this model. And we want to characterize these drift and diffusion processes, and we have built a hierarchical model for this, basically, where we have a Gaussian process priors for these drift and diffusion processes. And then we can, that of course regulates the complexity of the model and brings certain smoothness. Then by utilizing this framework, we can now aggregate the information across the multiple short time series, and we can recover the drift and diffusion even when we don't have this long continuous ones. And we have then validated this is still unpublished, but we have validated this with simulations from different models like Ornstein-Uhlenbeck process and a CASP model for more complex cases like BA stable distributions. And then we have applied this to our data from HIT-CHIP, which is human intestinal tract chip data with Willem de Vos, where we have this case of short time series on top of the population level thing. And we can recover quite nicely the expected properties of these time series here. Like the drift corresponds to what we expect to see for this B-stable Prevotel, for instance. And of course, you can then do this for many other sub-communities that are available there in the microbiome. So we are one step closer to characterizing the dynamics of this population landscapes when we do not have access to very good long dense time series. But of course, this was for a univariate time series. Often we are interested in analyzing more complex communities with multiple interacting partners. And we have been working with interaction models for community dynamics as well, where you can then model the interactions and the complexity that arises from that. There are many different models for community dynamics describing pairwise interactions. Some time ago with Caroline Faust and Didier Kontze, we were analyzing this kind of model where you can see it here. So we modeled the dynamical process as a function of growth rates and death rates as usual, and this is the function, growth function then. So interaction models in general yield more complex dynamics and alternative stable states, and we were analyzing this model and showing how different parameterizations of this model will lead to either discrete alternative states or more continuous gradients on this population landscape of the expected microbiota. And that was kind of part of this discussion about how the different landscapes emerge and whether there could be these enterotypes or alternative states of the gut ecosystem. This was kind of one way to look at this. When you have a more strong intergroup interactions and within the groups, when you have stronger interactions than between the groups, you can have these blocks between the different sub-communities, and then you can have subpopulations of individuals that then tend to cluster in different parts of the community space. But that was about modifying the interactions. But one problem with these kind of models, interaction models, is that they often ignore ecological memory. And this is one aspect of the interaction models we have been willing to promote lately. So ecological memory can arise from many different sources. So when species adapt to their environment, or there is heterogeneity, environmental modification, and so forth, there are different ways how memory can arise in ecosystems. But this is really often ignored in current mechanistic models of microbiome dynamics. This ecological memory is a quite well established thing in ecological theory and research. So that's surprising why it is not incorporated more. And of course, one reason might be that it's just difficult to do. So one way to do this is fractional calculus. There is, maybe some of you might be familiar with this, there is a long tradition in mathematics to do fractional calculus models. So basically, in differential equation systems, we typically use integer order derivatives, but there are also ways to use non-integer derivatives in these differential equation models, and those have interpretation as memory in the system. That has been shown by others. And so when you replace the differential equation models with these non-integral derivatives, and you can tune the level of how far it is from the integral derivatives. You can tune the level of memory by adjusting these parameters. So this way we can incorporate memory in the system. And we were doing this first for this concept model that I showed on the slides. So we were incorporating memory this way in this differential equation system, and then we were analyzing how the system is reacting to perturbations, different types of perturbations. So we subjected this simulated community to pulse perturbations, periodic perturbations, and stochastic perturbations, and we were analyzing how the incorporation of memory will influence the system dynamics. So there are different things we were looking at. Resilience was one of the first ones. So of course we can ask how memory influences the resilience of the system, and it's quite intuitive. When there is a small perturbation, small rapid perturbation, then the system tends to return quicker back to its original state when there is memory. But it's a little bit more complex than that, because also if you have insert memory in the system, here you can actually see this more rapid return to the original state when we add a little bit of memory. So one thing is that when you have longer time away from the original state, also the memory plays a different role, and it becomes more slow to return to the original state. So actually the effects of resilience can be twofold. It can depend a little bit on how rapid perturbation it is and how far away you are, how long time you spend away from the original state. So impact on the resilience is mixed. Memory can increase resistance. So here, this is what we had on the first slide. So this is community with no memory, and we add some pulse perturbation on the growth rates or memory of one species, the green species. So when we add the green species a little bit in the system, it tends to go towards state which has more green species, but then when the perturbation stops, then we return to the original state. And as you can see, memory slows down this. But now when we increase the perturbation, so we have the same perturbation, but it's a bit stronger now, then the system actually reaches a state where we can switch to another state when there is no memory. But when we add memory, the system is still more resistant to this state shift. So it doesn't shift that easily to the new state even after stronger perturbation. So memory can also increase the resistance and prevent state shifts in communities. When we have periodic oscillations, memory can make these oscillations stronger. There is a hysteresis effect, and this is maybe not so surprising, and also the same kind of thing can happen even when the oscillations become stronger. It may still prevent the state shifts in the system, and this can result in more long-lasting and transient oscillations. Maybe can help to understand some of the ongoing transients in dynamical systems like gut microbiome, but it also makes the system a bit more unstable during the transients. One aspect of this is that changes in memory properties can lead to also provide one mechanism that leads to alternative stable states. So in this earlier work that I mentioned, we were looking at how changing the interaction parameterization can lead to different alternative states in the system. But now, even when we keep those constant, and we alter the memory properties of the members of the community, we can also recover different alternative stable states of the system. Maybe not that surprising, but it's kind of nice to see that this provides an alternative complementary mechanism for alternative stable states. And probably in real systems, you can have mixtures of all these different things going on. Finally, we were also looking at this in an experimentally parameterized model. So Venturelli and others have recently quantified models of gut microbiome community dynamics, and they have measured some growth rates and interaction parameters for these species pairs from the gut ecosystem. So we just took those parameterizations from that paper to see if our model would also work well in this kind of two species communities parameterized by real data. And essentially the outcomes of our analysis with this data, real data parameterized model is the same than from our simulations. So the impact on the stability landscape on community composition, we have been testing different types of perturbations and community structures, two different types of models like this Concha model and generalized Lotka-Volterra, and different types of memory. Sometimes we tune the strength of the memory, but sometimes we also tune the how commensurate the memory is, whether all the species have similar level of memory or if they have differences in what level of memory they have. And we have shown multiple impacts on different aspects of community dynamics. There are, of course, many limitations and extensions. So this is so far based on simulations. So we still struggle with fitting this on real data. You can, of course, optimize parameters, but it is not, and it is often can be unidentifiable and so forth. So fitting these models to real data can be harder, but it is, of course, very relevant problem as well. And there are specific shapes of memory in this model. It's time decaying things, so things closer in the past will influence the system more than things further away in the past. So there is a particular decaying shape of the memory, but there can, of course, be many different types of memory, different shapes of memory, and this is just one way to model and incorporate memory in the system. There can be other ways, and maybe it can also depend on what type of communities and what type of ecological mechanisms you are looking at. And it would be, of course, interesting then to study this in more detail and analyze this in more extensively. We also have to show if there are performance gains in real applications based on this. So this was recently published in PLOS Computational Biology as collaboration with Moin Kalihi, who does PhD thesis in my team, Guillem Sommerier-Klein, who is here also in this workshop, Didier Concha and Caroline Faust. And then Giulio Benedetti from our team also built a Julia package together with Moin to do some of these simulations. So if you're interested in this, you can have a look at this Julia package. And then the time is soon over, but I wanted to say a few words about our methods development. So some of these techniques and tools we want to make more broadly available for the community. So we have been working a lot with R and bioconductor frameworks. So you probably have heard about bioconductor or even used it. So it's a global bioinformatics project that started about 20 years ago and already has tens of thousands of citations to different tools in this ecosystem. It's one of the largest bioinformatics projects in the world with more than 2,000 software packages. And one of the key things here is that there is heavy effort on standardizing the tools, making them more interoperable and reliable by unit testing and automated builds and checks and all those things. So of course, this is not applicable to all computational research, but whenever we make tools and methods that are more widely usable by the community, we can take advantage of this. And one important part of this is the so-called data containers. So we have standardized ways of representing microbiome data in R. Phylosec is one, and then there is an alternative way to do this tree summarized experiment, which helps you to build the abundance tables in the system, potentially with different assays that correspond to different transformations of the data. You can have side information for the taxonomic groups, like sequences or taxonomic classifications or information on their functional roles and so forth. You can have side information for the columns, which are the samples. So age, BMI, health status, geographical location of the sample, and so forth. We can also have hierarchical side information for the rows and columns. In the context of microbiome, this hierarchical side information would correspond to phylogenetic trees. In the context of the samples, which are on the columns, this would correspond to nested study designs, family relations, host phylogenies, and things like that. So that's a systematic framework for incorporating these many different diverse types of side information on top of your abundance tables and keeping everything very organized. If you subset or manipulate the data, it will deal with all this data object at once. And we have importers from all the common data formats like biome files or human 2 and 3 formats and data to other formats. So you can import data from standardized format to this data object in R, and then there is a whole ecosystem of methods and packages that can operate on this data object and provide you different tools for data visualization and analysis. We were speaking about this first time in European Bioconductor meeting in 2020 with those guys that you see here. And one aspect of this is MIA time package, which provides now some tools for microbial time series analysis. This is relatively small package still, but you can use it to visualize time series with confidence intervals, and you can also calculate very easily divergence from the baseline, for instance, and do different types of manipulations like this. In this example, we have a minimal cut dataset, which is like a 16 species system from a bioreactor, and we have here 3 different bioreactors followed over time, so you can calculate the divergence from baseline and visualize it easily along all these different bioreactors along the time. So it's a set of handy tools for this kind of analysis, and we are building now an online tutorial that is in beta version that is already online and giving different courses about how to use this framework for the analysis of microbiome data. So if you're interested, you can have a look at this online tutorial, and we are welcoming any suggestions on how to improve that further. So I think with that, I would like to close and just thank all my collaborators. Guillem and Chandler are here present as well. And we also have open PhD positions available in Turku and Finland, which is here in the southwestern Finland, quite close to Helsinki. It's the oldest academic city in Finland with about 200 inhabitants. So if you are interested in working with this kind of topics, welcome to contact me. Thank you very much, and that's it.
That was super, Leo. Loads of fascinating methods and results. So I'm sure there's some questions.
I didn't understand some of the aspects of the memory that you have in the model. So does it affect both the memory of sort of where the species would move to in the next time step, or does it also affect like how long it stays where it is before it moves anywhere? So like the waiting time distribution between assumptions.
Right. The memory influences, well, basically, of course, it's a general framework. We can look at different aspects, but here we were thinking about it in the context of microbial growth rates. So when in these interacting community models, we are looking at the abundance of the species. So the state is always the abundance of the species, and when the abundance is changing, it's increasing or decreasing, then this rate of increase or decrease will change when you add memory. So if you add memory, the rate of increase or decrease becomes slower.
So when you're fitting that, what sort of parameters do you have?
When we are fitting that, the parameter is the non-integer order of the derivative. In differential equation models, you typically use these integer order derivatives. So now we replace those with non-integer orders that are typically quite close to the original integer order in the model, but that's the parameter that we tune. So when you tune that parameter, then you are essentially incorporating memory in the model.
I'm confused because I thought yours had a kernel as well, or?
Sorry?
I thought there was a kernel or something. Yeah, maybe I misunderstood.
No, it's the shape of the memory from this kind of system becomes like a decaying, like.
I see. So it's sort of like an effective kernel.
Yeah.
My question is, do you need a special solver for the system?
Yeah, that's a good question. Yes, you need specialized solvers, but fortunately other people have been building those. In MATLAB, some quite efficient ones have been available, but recently there has also been development on this in Julia, also by others than us. But then we wanted to also build a solver, and there are now, for some cases of this model, there are now solvers in Julia. Yeah.
Could you give some insight into how it's different to modeling, say, fractional Brownian noise?
How, sorry?
So another way to model memory is to incorporate into a stochastic model and capture, you know, noise and then dependency through the stochastic term.
Yeah, we were analyzing stochastic perturbations in this model, and essentially like the same kind of thing that you see with more clear perturbations like pulse or periodic perturbations. Stochastic perturbations give like, I mean, the outcome are similar. Of course, the perturbations are more stochastic, but the outcome is the same. So the system becomes slower. The transients become more unstable, and like similar kind of outcomes apply to this system. One final thing I want to say about this landscape thing is one thing that is making very difficult to analyze this is that whereas in most typical interaction models, the landscape is kind of fixed and we can analyze the properties of the landscape. When you incorporate the memory in the system, the landscape is moving all the time together with this, this ball that is moving on the landscape. The landscape is also changing all the time because the memory is adding dynamics also on the landscape itself. So it will depend on where the ball came from, how the landscape looks like. So that's making it more difficult to analyze these systems formally.
Okay, if there are any more questions, I'll ask one quick question. So do you have any sense of the intuitive basis for these, the biological basis for these memory effects?
Yeah, right. So one is phenotypic plasticity, for instance, like in bacterial populations, for instance, you have a certain degree of variation, and when you have environmental pressure, of course, the system will evolve towards a certain type of community. And then even when, let's say, if we speak about antibiotic resistance, for instance, then if after you remove the perturbation, the system will keep that resistance for some time, but after a sufficiently long time, it will disappear. So that's one example of memory. But then, of course, resource accumulation, if the organisms change their environment somehow, that will also kind of, as a community, at the community level, you can think of that as a form of memory. There are many different mechanisms that could lead to memory in these kind of systems.
Oh, well, that's fascinating. Let's thank Leo again.
Thank you.
You got your talk later. Okay, well, our next speaker is Todd Parsons